however, you want to tear one down, if you want to tear a sugar up so that you can use it, for example, I want to use the glucose molecules to make energy. How do I isolate them from the larger molecules? That process is called hydrolysis, the removal of water. You take water away, you cut it up. Sorry, I said that backwards. You add water to the mix, you cut it up. You take water away, you build. So to dehydrate is to build organic macromolecules. To hydrolyze is to tear them down. So the important disaccharides for this class are sucrose, lactose, and maltose. So you can see maltose is built by putting two glucoses together, and lactose is built by putting the lactose and glucose together. So some of you may be familiar with the condition known as lactose intolerance. And so the enzyme that actually creates the hydrolytic reaction there is problematic for these people. And so they have a problem in breaking down this particular sugar. Okay, so for today, sucrose, lactose, and maltose, the disaccharides, and the monos that are used to construct them. Sugar molecules. Now, this is the movement from mono to disaccharides. But as you pointed out, the sugars that we ingest in our foodstuff are not normally disaccharides. They are polysaccharides, or things that we call carbohydrates. And they're the general plant molecule, as you pointed out, that it's the sugar-carrying molecule. The polysaccharide in plants is starch, and the one in animals is glycogen. So this is a picture here of a branched glycogen molecule with lots and lots of glucose units attached to it. So listen to the terminology. Monosaccharide, disaccharide, polysaccharide. Mono, di, and poly. These are common terms used to describe the formation of organic macromolecules. And what is the general chemical reaction that occurs as we build them? Dehydration synthesis. And if I want to tear them down, hydrolysis. Okay. Here's the second group of organic macromolecules. These are the triglycerides. Sorry, these are the lipids. Big category is lipid. We think fat when we say lipid, and that's good. But the lipids I want to introduce to you exist in the human body in three different primary forms. This is the first one. Lipids exist in the human body as neutral fats or triglycerides. Once again, I will not ask you to draw any specific triglyceride. However, the basic mechanism for putting it together and what is used to construct it, you will need to know. So if you had to guess, from what you just learned about sugars, what would be the simple chemical reaction that I would use to describe the construction of a triglyceride, an organic macromolecule? What would that be called? Dehydration synthesis, exactly. And so here's how the water thing works. A 3-carbon alcohol called glycerol has attached to it 3 chains of carbons. And in that process, water is removed in a process called dehydration synthesis, 3 times to form a triglyceride. What would happen if I cut one of these off? One of these fatty acid chains off? What do you want to call it then? A diglyceride. What if I want to cut 2 of them off? Then what do you call it? A monoglyceride. Right. Okay. So the monomeric units used to build triglycerides. Now let's point out a couple other things here. I put this one first on purpose because if you see sugar, so let me tell this young man because he's worried about the slides. All of these slides, he's brand new in the class. And he doesn't know how much I love him. They know. But you don't know this yet. The class is being recorded for you. And these slides are all on the portal already. So you don't need copies of them. When you come to class on Friday, you can print you off a set and bring them. So after class today, when you stop and talk to me, I'll show them to you. Okay, great. All right, so triglycerides, here's the reason I put them in first like this. Because The first inclination, if I say sugar, where does your mind go?
Energy.
Energy, right? Energy for body processes, making ATP. And that's where every book goes with it. You take the glucose molecule and you run it through the 10 steps of glycolysis, and off you go in making energy. That's how every book does it. Now, this semester you will learn about skeletal muscle contraction. This semester. I'm going to teach you the physiology of skeletal muscle contraction. And as I'm walking through it, I'm going to identify 4 huge areas, 4 primary reasons why muscle contraction will not happen without energy. And then I'm going to show you a graph that's not in your book. And that graph is going to tell you that muscles, skeletal muscles, and cardiac muscle actually prefer to use the carbons of triglycerides for their energy over against the carbons of sugars. Now, the simple reason for that, the simple explanation for that is there are many, many more carbons available in a triglyceride than there is in a sugar molecule. How many carbons are available in a molecule of glucose? 6. How many carbons are available in any given triglyceride? That dot, dot, dot right there means a lot more goes here of the same. CH2, 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 CH2. Are you following me? Lots and lots of carbons here to make these triglycerides. So, gram for gram, this stuff has a lot more energy in it, because energy in the human body is carbons. Cutting up carbons. That's how you make energy. You cut up the carbons. All right, so there's a lot more carbon available here to burn for energy in triglycerides. Now, the other really popular way to talk about triglycerides in our society today is to talk about saturated and unsaturated fats. So this picture here, with these 3 fatty acid chains here, is a picture of a prototypical, completely saturated fat, very common in animal tissues. Saturated fats. When you say saturated to a chemistry person, what you mean is all of the bonds on the carbons are bonded to either carbons next to them or hydrogens. So there are no double bonds. So each carbon, this carbon is attached to this one, and 2 hydrogens, 2 hydrogens, 2 hydrogens. So the full complement of 4 bonds on the carbon are there. These are saturated carbons. You see it? They're saturated with 4 bonds each. Now, we know now, because of the way the body processes fats, that carbons, triglycerides that have a higher concentration of unsaturated carbons tend to be a little better for the lining of your blood vessels. We call those fats unsaturated because hydrogens have been removed, and the electrons are actually sitting between the carbons. So when you look at a triglyceride and you see all single bonds, those are saturated fats. When you look at a triglyceride and you see double bonds between the carbons, those are unsaturated fats. And the more double bonds you see, the more unsaturated the fat is. Now, it's going to be a while before I can teach you more about that. We'll get all the way to the cardiovascular system before that will come up again. But I want to point it out here as a take-home message for you to remember. Triglycerides. It is fascinating. I get distracted with these things. It's fascinating to me how the manufacturers of the products that we ingest, that you get in the grocery store, get around federal regulations. Whenever your body uses a triglyceride, it will process this molecule differently than any of the others. All the other organic macromolecules, when you cut it up to the monomer and you absorb it through the intestinal wall, The intestine takes it by way of the capillaries to the liver for further processing. Well, when your body ingests fat, triglyceride in particular, after the molecule is broken down, how would you do that? It's not dehydration synthesis. You would need to do hydrolysis. You got to cut this thing up with hydrolysis. So you need enzymes to do this with. And once you've done that and cut it up, and it enters into the cell of the intestinal wall, the intestinal wall actually rebuilds it into a triglyceride and then coats it with a protein and then ships that thing out as though the blood vessels are supposed to take it, and they cannot. So instead, a different group of capillaries take it up, called the lymphatic capillaries, the lacteals, and all the fat goes directly into circulation at the subclavian veins, right at the heart. So the triglyceride that we ingest doesn't get processed in the liver first. The first place it goes is to the blood vessels in the heart. And so students learn this and they go, that's crazy. But is it? Is that crazy? So tell me about take-home message. Where is there more energy? Sugar or fat? Gram for gram, where's there more energy? In the fat. Why is there more energy in the fat? More carbons. Why would that matter? Why would your body want to take these molecules to your heart directly? What?
Because your heart's not working?
Yeah. I mean, of all the muscles in your body, this one has to contract. Every cell in the muscle mass has to contract every second of your life. In terms of energy needs, it'd be hard to find another structure that needs more. So the whole thing is designed that way. Take the high energy molecule to the organ that needs it. I find that kind of stuff flabbergasting. Is that a word? Flabbergasting? I don't know. Okay, here's the second one, and I've got this one drawn on the board. This is the second lipid. These are the phospholipids of cell membranes. So over here you see some things that you recognize. First of all, there's my 3-carbon backbone. You see it, the glycerol? And it's attached to. So if I just drew this and this, what would you call this molecule? Just the yellow part. What would you call this? I like it, a diglyceride. Saturated or unsaturated? Unsaturated. You see, did y'all find the double bond right there? Okay. Okay, so, but that's not the way we teach phospholipids. That's just my way of saying, look, I just taught you this. That's not what this is. This is not a triglyceride, and it's not a diglyceride. Instead, these very non-polar, uncharged, non-charged, loving portions of this molecule are attached to a phosphate or phosphorus containing, you see the charge on that guy? This part of the molecule, the phosphate head, which is illustrated there, is a charged molecule. Now the beauty of this for phospholipids is organelles within the cells can actually produce these. They're made in the smooth ER all the time because we're damaging our cell membranes all the time. So you have to rebuild them. But you don't need any special mechanism to put them together. So over here I've drawn the way they're oriented. In order to construct a cell membrane, you have a polar charged head oriented out and oriented in, and non-polar fatty acid tails on the inside. There is no mechanism, there are no protein carriers or any other sophisticated network used to build it like this. They will self-assemble. And the reason they will is because this environment and this environment is water. Water. Dihydrogen oxide, the most toxic, y'all don't know the joke, I'm not going to tell it to you. H2O, right? And the hydrogens carry slight positive charge, and the oxygens carry slight negative charge. Water is a polar molecule. If you put it in something, it will tear it up. You put your dirty pot in the sink, put some water in there, right? And let the polar molecule go to work. This